everyone present here. Myself, Navjeet Kaur, on behalf of Amar Shahid Baba Ajit Singh, Jujhar Singh Memorial College of Pharmacy, Bela, extend a warm and respectful welcome to all our dignitaries, esteemed speakers, authorities, faculty members, and dear students. Today marks day one of our popular lecture series sponsored by the Department of Biotechnology, Ministry of Science and Technology, Government of India. We are deeply honored to have among us our distinguished guest and speaker for the day, Dr. Anup Kumar Sir from Delhi Pharmaceutical Sciences and Research University, New Delhi. First of all, I request Director Sir Rahul Kumar Sir kindly offer a floral welcome to our today's speaker, Dr. Anup Kumar Sir, and our guest of honor, Dr. Sabinder Kaur Ma'am. It's a privilege to welcome you to this academic celebration of science and health. Thank you, sir. To begin this enlightening journey, let us invoke the divine blessings of knowledge and wisdom. I request Director Sir, Organizing Secretary Dr. Rahul Kumar Sharma Sir, and our eminent guest of honor Dr. Satvinder Kaur Ma'am, and our honored speaker Dr. Anup Kumar Sir to please kindly come forward for the lamp lighting ceremony. May this light guide us toward new discoveries and meaningful learning. Firstly, I would like to invite our respected director sir to deliver the welcome address and to share a few words about the institution and this noble initiative. Please give them a huge round of applause. I request the dignitaries kindly take your seats.
Very good morning to all of you. आवाज नहीं आई. So, our respected today's guest of honor, Dr. Satvinder Kaur Madam, ABGI Women Forum, and Principal GHG Khalsa College of Pharmacy, Gursar Sudhar. Today's first expert of the popular lecture series, Dr. Anup Kumar Sir from IFSAR, New Delhi. Organizing Secretary of the program, Dr. Rahul Kumar Sharma ji. Coordinator of the program, Dr. Ajay Singh Kushwaha ji. HODs, faculty members, non-teaching staff, and my dear students. Today, as we start a DBT-sponsored program, Department of Biotechnology of Ministry, Health and Sciences, Science and Technology, गवर्नमेंट ऑफ इंडिया, ये जो सीरीज है इसके अंदर 3-4 लेक्चर्स किसी एक टॉपिक के ऊपर पूरे होंगे और जो इस बार टॉपिक लिया वो था एंटीबायोटिक रेजिस्टेंस से रिलेटेड। तो जितने भी आज हमारे स्पीकर्स होंगे वो सब इस पर बात करेंगे। इसकी जितनी भी अभी तक की नई नई चीजें खोज हो रही हैं उन सबको आपसे साझा करेंगे। तो इट इज वेरी मीनिंगफुल टू ऑल ऑफ अस, हम इससे पूरा ज्ञान लें, सीखें। जो चीज समझ में ना आए उसके एक्सपर्ट हमारे पास हैं तो उनसे जरूर उसके बारे में क्वेश्चन करें, अपनी क्वेरीज को रिजॉल्व करें। सभी को बेस्ट ऑफ लक और सीखने के लिए। साथ में मैं यहाँ पर धन्यवाद भी करना चाहूंगा डिपार्टमेंट ऑफ बायोटेक्नोलॉजी का, जिन्होंने ये अवसर भी प्रदान किया हमें स्पॉन्सर करके इस प्रोग्राम को कॉलेज में ऑर्गेनाइज करने के लिए। तो थैंक यू वेरी मच ऑल दी गेस्ट स्पीकर्स एंड पार्टिसिपेंट्स जिन्होंने यहाँ पर आके इस प्रोग्राम को ज्वाइन किया। जय हिंद, जय भारत।
Thank you, sir. Now I would like to invite Dr. Satwinder Kaur ma'am. Please come forward and say a few words. Thank you. A very good morning, all of you. Greetings of the day. First of all, I would like to thank Dr. Satwinder for giving me this opportunity today to talk to you, all of you directly. So, I congratulate all of you that you have got a very dynamic principal, Dr. Sai. His energy and his outlook and his visions are really commendable. And I congratulate all of you that you are working under his guidance. At the same time, this antibiotic resistance is very important for everyone. There is so much of indiscriminate use of drugs these days. And if you compare, being a pharmacist, you must have noticed that in other countries, in foreign countries, antibiotic is not prescribed as such. They don't give anything for violence. They say you go home, stay for 14 days, and you will recover. Emergency is only dealt with. So, personally, after we only going up and living in foreign, very close relative or something, have such cases. So, in India, this indiscriminate use is really to be addressed. So, I congratulate the organizers for choosing this topic. At the same time, the speaker for today, Dr. Anup Kumar, he is there to solve your queries and well-versed dignitary and a speaker for the topic. And I also congratulate Dr. Rahul, Dr. Deshmukh, Dr. Kushwaha, and Madam Monica for being a part of the team. And I wish you all the very best and hope you will learn a lot and increase your knowledge from the series of lectures, which is being organized by COPBELA College with support of DBT. Congratulations all and happy. Thank you, ma'am. Now I request Dr. Rahul Kumar Sharma, sir, organizing secretary of this event, to kindly address the audience and brief us about the objectives and vision behind the DBT-Supported lecture series.
गुड मॉर्निंग एवरीवन। मैं सभी को मेरा नमस्कार। आज मैं माँ सरस्वती के चरणों में वंदन करते हुए इन 3 दिनों की डीबीटी स्पॉन्सर्ड लेक्चर सीरीज के बारे में अपने विचारों को प्रकट कर रहा हूँ। And we are delighted to welcome you to DBT sponsored popular lecture series organized by Amar Said Baba Ajit Singh Jawasi Memorial College of Pharmacy, Bela Road, Punjab, from 9 to 11 October 2025. This academic initiative has been carefully planned to provide a platform for students, research scholars, and faculty members to engage with renowned experts and gain exposure to the latest advancements in immunology, antibiotic resistance, and growing challenges of the superbugs. Through this series, the eminent speakers from Dexaru, IIT BHU, Punjab University, and our college also will share their rich experience and scientific knowledge and current research insights. The program will not only broaden your understanding of critical biomedical issues, but also encourage the interaction, discussion, and exchange of innovative ideas that are essential for academic and professional growth. Active participation in these sessions will inspire you to pursue the new dimensions in your learning and research journey. Once again, we sincerely thank you for your participation. We hope this lecture series will be memorable and enriching academic experience that lead add value to your future endeavors. Thank you and keep learning always. Thank you.
Thank you, Dr. Rahul. Now I request Director Sir and the Organizing Secretary kindly felicitate Dr. Supinder Kaur Ma'am with a token of love and respect.
Madam.
Thank you, sir. Now it's time to meet our eminent speaker for today, Dr. Anup Kumarji. It's my privilege to introduce today's distinguished speaker, Dr. Anup Kumar Sir, Assistant Professor, Department of Pharmacology, Delhi Institute of Pharmaceutical Application and Research, New Delhi. Dr. Kumar is an eminent researcher with over 12 years of experience in teaching, research, and industry. His key areas of expertise include pharmacovigilance, drug safety, network pharmacology, and antimicrobial drug discovery. He has authored more than 100 research publications, several book chapters, and books with Springer and CBS publishers with over 2,900 citations. He has successfully handled DST, SERB, and ICMR funded projects and was recognized among the world's top 2% stances by Stanford University in 2021. Dr. Kumar is also an active member of APTI and IPS and contributes to various academic and editorial boards. We are truly honored to have him with us today to share his valuable insights under the DBT-Supported popular lecture series on the theme antimicrobial resistance. Stage is all yours.
Hello. So, very good morning, everyone. Good morning. So, first of all, I would like to thank Salesh sir, Ajay sir, Rahul sir, all the faculty members of DELA for giving me the opportunity to be part of this popular series. Thank you so much. And for today, my topic, which is allotted to me, is related to immune systems. I have worked with respect to immune system, particularly with thymocytes and spherocytes. So I am going to start with very basic, what actually the immune system is and how immune system works. I hope many of you already knows what is immune system, how it works. But purpose is, if you want to conduct a experiments related to immune system, so how you can plan and how you can conduct these kind of experiments with help of computational techniques, how you can explore the immune system. That's the one part. Another part, by doing the in vitro studies, you can do the culture of thymocytes, phenocytes, and by conducting the animal experimentation. So these are the three aspects: computational studies, in vitro studies, and in vivo studies. So if any one of you plan to do some assays or some, so you want to check the effect of chemicals or drugs on the immune system, so how you can do it. So again, just starting from the basics, I hope every one of you knows that. Can anyone answer how many types of immune system? Broadly, we are classifying into how many types and what are these? Anyone? I hope it should be interactive. Anyone? Immune system कितने तरह का है generally? And immune system is important. Why this lecture is with respect to immune system? Because when we talk about microbial agents, pathogens, so first thing which comes in a play is immune system, right? So anyone can answer. Immune system is of. Also, to say आपको मैंने please बहुत तरह का. Can anyone answer how what are these two types? Very good. Adaptive and innate, right? You can call it active, passive as well. So there is a innate immunity and adaptive immunity, right? So what is innate immunity? As its name indicates, innate. जबकि आपको whenever you are getting to be infected by the pathogen, the first immune system which plays a significant role is innate or adaptive. Innate immune system. That's why its name is innate. Whenever any pathogen enters in your body, the first system which will act is innate immune system. So under innate immune systems, there are in body there are three lines of defense: primary defense, secondary defense, tertiary defense, third line defense. So primary and secondary defense makes your innate immune system. What comes under primary defense? Primary defense, your skin and mucous membrane. That is comes under your primary defense, right? So if you see under microscope in this room, you will find there are a lot of microorganisms are there. So which part of the body is preventing the entry of microorganisms? So which part? Skin. Skin is a barrier. Skin is a barrier for the pathogens which can enter. Otherwise, the pathogen will enter inside the body, right? So skin contains a protein. That protein name is, quickly answer this, keratin, right? Skin contains a keratin proteins which prevents the entry of microorganisms inside the body. But microorganism can be entered. Just I am talking, there is a, we are, inhaling air. So microorganism can enter through nose, microorganism can enter through mouth as well, right? So again, there is a mucous membranes which will release the mucus, which will release the enzymes, which will do the degradations of your pathogen. Once a pathogen enters into your GI tract, you all know in the stomach there is a acid. So acid will try to kill it.
And if your pathogen is so strong, if it's not killed by all these mucus or enzymes or your acid, then it will going to enter into your systemic circulation, right? So in systemic circulation, which cells play immunity? Anyone answer quickly? ये सबको पता है, बताओ. White blood cells, WBC are the major cells which provide the immunity. So these primary WBC provide a secondary line of defense. So whenever any pathogen enters in our body and reaches in the systemic circulation, so in your blood there is a WBC which will try to kill that pathogen, right? So that comes under secondary line of defense. And primary and secondary line of defense makes your innate immune system. Is it clear? And then there is a last में दिखेगा बोर्ड लास्ट वाले स्टूडेंट को maybe okay. So third line defense, under third line defense is your T and B cell. How many of you heard about T and B cells that provide specific immunity to the body? Specific immunity, and that we call third line of defense. So there are three line of defense: primary line of defense, secondary line of defense, and third line of defense. So primary and secondary line of defense makes your innate immune system, and third line of defense, that is your T and B cells. These are highly specialized immune cells which provide specific immunity, and that immunity we call adaptive immunity. And why we call it adaptive? Anyone? Innate, I told you. Innate immunity, why we call it innate? Because this is the immunity which at first sight, this is the immunity which is going to occur. But why it's adaptive? What is the meaning of adaptive? Anyone? Okay, very good. Any other one? Adaptive. What is the meaning of word adaptive? Adaptive means it will take time, right? It will take time to adapt. So that's why whenever any pathogen enter in our body, so your T and B cell is not going to play a role at initial phases. Initial phases, your innate immune system will try to handle it, but when your innate immune system is unable to handle, then that pathogen will hand over to your adaptive immune system. And during this process, it will take 7 days or 5 to 7 days. आप में से कितने लोगों को चिकन पॉक्स वगैरह हुआ है? Raise your hands. चिकन पॉक्स में आपने कोई दवाई ली फिर? दवाई खाई थी कोई? डॉक्टर ने क्या बोला? टाइम लगेगा. क्यों लगेगा टाइम? बताओ क्यों लगेगा टाइम? हाँ, इम्यूनिटी, क्योंकि अडेप्टिव इम्यूनिटी को एक्टिवेट होने में टाइम लगेगा. क्योंकि अडेप्टिव इम्यूनिटी को आपके 5 से 7 दिन लगेंगे एक्टिवेट होने में. And T and B cells are powerful cells which provide the specific immunity, right? So these are just introductions about your immune system. One is primary line defense, secondary line defense makes innate immune system, and your T and B cells makes your specific immunity that we call adaptive immune system as well. So innate immunity we also call non-specific immunity, and your adaptive one we can also call it a specific immunity, right? But major function of immune system. How many of you heard the news about Nobel Prize? Nobel Prize recently given in the field of medicine. How many of you have heard about it? And for what discovery the Nobel Prize has been given? Anyone? Okay, not antibiotic. Anyone? आपने न्यूज पढ़ी है नोबेल प्राइज जो अनाउंस हुआ है मेडिसिन का, उसमें क्या डिस्कवरी की गई है? किस डिस्कवरी के लिए मिला है? एनीवन? इम्यून सिस्टम से रिलेटेड रिसर्च है, that we will discuss in slides as well, that is related to your immune system and what discovery they have done and how that discovery could play a significant role in drug development and discovery. That we will also discuss. So what I mean to say, the major function of immune system is when your pathogen enter inside your body, the major function of your WBC should have the ability to recognize between self and non-self. That's very important. If, for example, your pathogen enters, आपका कोई भी pathogen can be bacteria, can be virus, can be parasite, whatever it is, right? आपका pathogen enters in the systemic circulation, If your WBC is not able to recognize, it's a non-self. So how your immunity will be, how your immunity signaling pathway is going to activate? It will not going to activate. So first major work or major thing is your immune system should have the ability to differentiate between self and non-self self. अगर आपका इम्यून सिस्टम यही रिकॉग्नाइज नहीं कर पाएगा कौन अंदर का है कौन बाहर का है तो कैसे काम करेगा? अगर उसने गलत रिकॉग्नाइज कर लिया, योर इम्यून सिस्टम हैज रिकॉग्नाइज जो सेल्फ है उसको नॉन सेल्फ समझ लिया, इट विल डू द रिस्ट्रिक्शन एवरीवेयर एंड दैट वी कॉल ऑटोइम्यून डिसऑर्डर. Autoimmune disorder in which your immune system is unable to recognize its own body cell, right? So first thing is your immune system should have the ability to differentiate between self and non-self. So how these has been done? So on your WBCs there are a sensor proteins. Means there are a protein which acts like a sensor, and that proteins will help to differentiate between self and non-self. On WBC there are a protein. For example, this is your WBC. So on WBCs, there is a protein, specialized protein. And what is the function of this protein? This protein will differentiate between self and non-self. It will recognize which one is self, which one is non-self. So if your pathogen enter inside the systemic circulation, it should be self or non-self? बताओ. अगर कोई पैथोजन आपकी बॉडी में एंटर किया सिस्टमिक सर्कुलेशन में, वो सेल्फ होगा या नॉन सेल्फ होगा? नॉन सेल्फ, राइट? तो योर दिस प्रोटीन जो डब्ल्यूबी से ऊपर प्रोटीन है, दैट प्रोटीन शुड हैव द एबिलिटी टू डिफरेंशिएट बिटवीन सेल्फ एंड नॉन सेल्फ, एंड दैट प्रोटीन इन साइंटिफिक लैंग्वेज वी कॉल इट PRR. PRR. That protein we call PRR. PRR is pattern recognition receptors. So as its name indicates, pattern recognition receptor. These are the receptors which will sense particular pattern. And where is that pattern? Pattern is on your pathogen. And where these proteins are? These proteins are on WBC. And what is the function of this protein? This protein function is to differentiate between self and non-self. Right? If someone, if someone have a problem with PRR, कोई पेशेंट्स है, उस पेशेंट्स में पीआरआर रिकॉग्नाइज ही नहीं कर पा रहा, सेल्फ एंड नॉन सेल्फ, so it's going to be failure. It's going to be, if it's recognizing, so PRR is pattern recognition. करेंगे, इसको रिकॉग्नाइज करेंगे, बताओ. नॉन सेल्फ को हम बोल सकते हैं, तो पैथोजन को रिकॉग्नाइज करेंगे. फॉर एग्जांपल दिस इज योर पैथोजन. ऊपर भी एक पर्टिकुलर पैटर्न है, राइट? दैट वी कॉल PAMP. PAMP is pathogen associated molecular pattern. PAMP किसके ऊपर है? पैथोजन के ऊपर. तो इसका फुल फॉर्म क्या हो? पैथोजन एसोसिएटेड मॉलिक्यूलर पैटर्न. दिस इज ऑन पैथोजन, राइट? अब दिस PRR should have the ability to sense this PAMP, right? तो जो ये पैथोजन एसोसिएटेड मॉलिक्यूलर पैटर्न है, so PRR should have the ability to sense it. So once it senses it, so in PRR, you might heard about, how many of you heard about TLR? TLR. कितने लोगों ने सुना है? रेडियो एंड TLR with respect to immunology. What is TLR? Toll-like receptor. So that is the one of the example of PRR. Toll-like receptors are a sensor. कहाँ पे है? On your immune cell, which will differentiate between self and non-self. Toll-like receptors are further categorized into 1, 2, 3, 4, 5, 6, like 1 to 11. And particular toll-like receptors, some toll-like receptors will sense viral protein only, like TLR3. Some will TLR4 will sense only bacterial protein. Some will sense lipopolysaccharides only. So there are different types. So PRR, one of the classical example is TLR, toll-like receptor. तो अगर कोई बात करे toll-like receptor की, so you should have the ability कि toll-like receptor एक सेंसर है जिसका काम क्या है? किसके ऊपर है सेंसर? इम्यून सेल्स के ऊपर, राइट? और इसका काम क्या है? सेंस करना पैथोजेंस को, राइट? तो toll-like receptors, कुछ toll-like receptors जो बैक्टीरियल पैथोजन को सेंस करेंगे, कुछ वायरस को करेंगे, कुछ फंगाई को करेंगे, राइट? तो दिस इज द toll-like receptors. And there is a NOD as well, node-like receptor. Node-like receptor is another type of PRR, node-like receptor, NOD, right? So these are under PRR. So as I said, the purpose is to sense this pathogen associated molecular pattern. Or it sense, for example, now your WBC is okay, you are a healthy person, your WBC sensor अच्छे से काम कर रहा है आपका, तो उसने इसको सेंस कर लिया, right? So what will happen next? Now your WBC TLR has sensed that this is non-self. Now what is next? Next is it's going to activate TLR dependent signaling pathway or TLR independent signaling pathway. So it's going to activate intracellular signaling pathway. Whatever things are happening, it's happening on the surface of the cells. Recognition जो हो रही है वो कहाँ पे हो रही है? Surface पे हो रही है. अब जब your WBC has recognized it's a non-self, it's going to result in activation of intracellular signaling pathway. So intracellular signaling pathway is going to activate, and now what is the purpose of the WBC? To kill this pathogen, which they have sensed, right? The pathogen is going to kill by the WBC. So there is a process you might heard about phagocytosis. How many of you heard about phagocytosis? What is phagocytosis? Yes. बता दो फटाफट. Phagocytosis. Yeah, good. Good. So pathogens को engulf करेगा, right? And it fuse with the with vesicles which contains the lysosome. It do the formation of phagolysosome and do the destruction of that pathogen. इसको engulf कर लेगा. There is a phagocytosis where cells, WBC cells are engulfing the pathogen. But engulf तब तक नहीं करेंगे जब तक वो सेंस नहीं करेगा ये नॉन सेल्फ है. अदरवाइज वो ये मैकेनिज्म आपका एक्टिवेट होगा ही नहीं, right? When your WBC sense that it's a non-self, now WBC is going to phagocytosis, right? Phagocytosis. So it will engulf that, and inside there is a vesicle which contains a lysosome indent. So this is the one of the way by which your bacteria or virus will be killed by innate immune system. That's the one way. Another way is without enzymes involvement, bacteria can also be killed by your oxygen-dependent killing. Oxygen-dependent killing means it is going to generate free radicals. You might have heard about free radicals. So free radicals can do the damage on this pathogen. So ultimate goal of your innate immune system is to destroy this pathogen, which is sense, right? Another pathway which is going to activate is like NF-kappa B and MAP kinase. You might heard about. How many of you heard about NF-kappa B and MAP kinase pathways? Yes, what it is? NF-kappa B and MAP kinase. MAP is mitogen activated protein kinases. So these NF-kappa B and MAP will act on the nucleus and do the transcription and translation process. And during this process, your protein will be synthesized, your cytokines will be synthesized like interleukin. You might heard about interleukin. Interleukin पढ़ा है? So interleukin, these chemicals will be released like interleukin, and purpose is to kill this pathogen, right? So purpose is to kill that pathogen. So to kill that pathogen, right? So दो तीन mechanism है by which your innate immune system will try to kill the pathogen which is sensed as a non-self. इतना तक समझ आ गया सबको? Anyone who has doubt on this? एक mechanism हो गया, उसने engulf किया lysosome के साथ, fuse हुआ और lysosome ने उसका destruction कर दिया, right? Another mechanism is there is generation of free radical, and generated free radical will do the damage on the bacteria or virus, whatever it is. Another mechanism is it's going to activate intracellular signaling pathway. By that one, your MAP kinases or NF-kappa B molecule will act on the nucleus and do the transcription and translation process. I hope you know transcription is DNA to or RNA to protein क्या है? Translation. So protein will be synthesized, these chemicals will be synthesized, and purpose of these chemicals is to kill this pathogen, right? So to kill this pathogen, purpose of these chemicals which is released to kill this pathogen, right? These released chemicals is also going to activate other pathways. Have you heard about JAK-STAT pathway? JAK-STAT pathway. So what are the ligand for the JAK-STAT pathway? Ligand क्या है for JAK-STAT pathway? Your cytokines, growth factors. So cytokines like interleukin can also activate your JAK-STAT signaling pathway. So sometimes if you go through the research studies, researchers have explored the role of JAK-STAT pathway in immune system. So the JAK-STAT pathways, I hope you know there are 4 types of receptor we talk about. GPCR, your ligand-gated ion channels, your nuclear receptors, and another one is enzyme kinase receptor. So this JAK-STAT signaling is related to which one? किससे related है? JAK-STAT जो signaling है that is related to enzyme kinase linked receptor, GPCR, ligand-gated or nuclear? Enzyme kinase. So in enzyme kinase, if you see the configurations of that receptor enzyme kinase, when there is no ligand bind with the extracellular domain of the enzyme kinase, it's in the monomer form, right? It's in the monomer form when there is no ligand bind. So these are monomer forms. When ligand bind in the extracellular domain, like your cytokines which is released, like interleukin, right? So what will happen? Your monomer will become dimer. So dimer is the active form of enzyme kinase, and when it becomes dimer in intracellular domain, there are a which molecule? JAK. JAK is Janus kinases. Janus kinases will do what? Janus kinases will do the phosphorylation of what? Phosphorylation of tyrosine. And phosphorylation of tyrosine will create the active sites for which molecule? Second molecule, STAT. STAT is, can anyone expand STAT? STAT का expansion करो. Signal transducer activator of transcription. Signal transducer activator of transcription. So its name indicates signal transducer. ये signal को amplify करेगा, right? And activate what? Activate transcription. और जब activate transcription होगी तो आपके protein synthesized होगी, right? So more cytokines will be released and ultimately going to kill your pathogen. That's the one. Another, there is a and we have a JAK inhibitors. We have a drugs which is going to block Janus kinases. Can anyone name any drugs? कोई भी दवाई का नाम बताओ जो JAK को inhibit करती है and is in the market, particularly for the treatment of cancers, autoimmune disorders. COVID में भी हमने यूज किया है. Baricitinib, the drugs which end with the nib. Baricitinib, have you heard about these drugs? There are drugs which are under clinical trials, which are STAT inhibitors. STAT are further STAT 1, STAT 2, STAT 3, STAT 4, like that. These are the molecules. So we have a drugs under clinical trial which is going to inhibit the STAT, right? When you want to slow down your immune system, for example, your immune system is overactivated, so you want to slow down, so you can give these kind of drugs, right? Then another pathway you might heard about, lot of research on these pathways with respect to immunology and other aspects as well. Have you heard about PI3K, AKT, mTOR pathway? What it is? PI3K, AKT, mTOR. Have you heard about these pathways? And these pathways also playing a role in the immunology. What it is? PI3K, can anyone expand what is the PI3K? इसको expand करो. PI3K. Phosphoinositol 3 kinase. AKT also known as phosphokinase B. mTOR is, बताओ, expand mTOR. We have a drug, rapamycin is an mTOR inhibitor. We are using it, right? So mTOR is mammalian target of rapamycin. Very good. तालियां बजाओ इसके लिए. So mammalian target of rapamycin, right? So if you compare this JAK-STAT signaling and PI3, AKT, mTOR pathway, you will find similarity. What is the similarity? These both are enzyme-linked kinase receptors. Which these pathways belongs to enzyme-linked kinase receptors. I already told you enzyme kinase linked receptor की configurations क्या है? It's in monomer inactive state when there is no ligand bind. When ligand bind, so this monomer will be converted to dimer, right? In dimer, in this JAK stack case, which one is the intracellular molecule? JAK. JAK is the intracellular molecule. But in this case, intracellular molecule is PI3K. Simple. Rest of the configuration is same. Only intracellular molecule is different. In JAK stack signaling pathway, intracellular molecule is JAK. PI3 AKT mTOR signaling pathway में intracellular molecule क्या है? PI3K, right? This PI3K, when your ligand binds, this phosphodiesterone 3 kinase is going to activate. It's going to do the phosphorylation of PIP2 into PIP3. PIP2 will be phosphorylated into PIP2 to PIP3. PIP2 is phosphoinositol diphosphate and PIP3 is phosphoinositol triphosphate. So who is going to do the phosphorylation? Your PI3K. What is PI3K? PI3K is phosphoinositol 3 kinases. Right? Further, this PIP3 is going to activate which is next molecule AKT. AKT is also known as phosphokinase B. And which is next molecule? Your mTOR. There is a FOXA as well, there is another molecule as well, but you can understand up to this mTOR, and mTOR has a role in cell differentiation. When there is any bacteria enters, you want more number of cells, more number of immune cells, right? So this is not PI3K. Now we have a drug which is going to block this. We have a drug which is going to block this. Which is going to block this, right? And when you want to block these pathways, कब block करना चाहोगे? जब immunity कम हो या immunity ज़्यादा हो? बताओ. Immunity excess हो. Autoimmunity immune disorders हो, right? So this basic is clear about innate immune signaling pathways. Any question up to this? Any question last में? समझ आ गया आपको? Clear? Right? So these are your innate immune signaling pathways. Now, you know that can every type of pathogens can be handled by the innate immune system? Yes or no? कोई भी वायरस बैक्टीरिया सारा इनेट इम्यून सिस्टम हैंडल कर लेगा? नहीं करेगा. कुछ आपके फॉर्म भी होंगे जो कि योर इम्यून सिस्टम इज नॉट एबल टू हैंडल दैट पैथोजन, राइट? दैट मींस दैट पैथोजन इज मोर स्ट्रॉन्गर, राइट? सो इन दैट केस व्हाट हैपन? इन दैट केस योर इनेट इम्यून सिस्टम विल कैच दैट पैथोजन एंड हैंड ओवर टू अडैप्टिव इम्यून सिस्टम. इनेट इम्यून सिस्टम बोलेगा मेरे बस का काम नहीं रहा है, इसको पकड़ के अगले उस पे डाल दो, नेक्स्ट अथॉरिटी के पास सेंड ओवर कर दो, राइट? सो दैट इज योर अडैप्टिव इम्यून सिस्टम. सो व्हेन योर इनेट इम्यून सिस्टम इज अनेबल टू हैंडल द पैथोजन, सो इन दैट केस योर इनेट इम्यून सिस्टम विल कैच दैट पैथोजन एंड हैंड ओवर टू द अडैप्टिव इम्यून सिस्टम. बट दिस इज a specialized mechanism. Immune system is very complex, you know. It's a specialized mechanism by which you have to hand over that pathogen to the adaptive immune system. Because problem is if your adaptive immune system अगर गलती से wrongly वो activate हो गया तो it will do destruction everywhere in the body. तो आप फिर उसको control नहीं कर पाओगे, right? So this is a specialized mechanism by which your innate immune system will hand over the pathogen to the adaptive immune system. So how it happens? So you know how it happens? Who will catch the pathogens? जो हैंडल नहीं हो रहा, जो बहुत डिस्ट्रक्शंस कर रहा है, इनेट इम्यून सिस्टम ने जोर लगा लिया अपना पूरा कि भाई मैं इसको किल करूं पर वो उल्टा मार दे रहा है. तो उस केस में क्या करना पड़ेगा? कौन इसको कैच करेगा? इनेट इम्यून सिस्टम सेल नहीं करेंगे, लाइक WBC sometimes your dendritic cells or other cells. These cells we call these cells which catch the pathogens we call APC. You might heard about with respect to immunology APC word. What is APC? Antigen presenting cell. So as its name indicates, they are presenting. What they are presenting? Antigen. किसको प्रेजेंट कर रहे हैं? किसको प्रेजेंट कर रहे हैं? अडैप्टिव इम्यून सिस्टम को. और अडैप्टिव इम्यून सिस्टम का पार्ट्स क्या है? T and B cells. So particularly they will hand over this pathogens. ये इन्होंने कैच किया pathogens. But अब इसमें specialized mechanism, what kind of specialized mechanism is? These pathogens will be catched by specialized proteins, which is present on the cell surface of your antigen presenting cell. And that protein we call, you might heard about MHC. Have you heard about MHC? MHC, major histocompatibility complex. आपने ये तो सुना होगा ना कि जैसे मान लो किडनी डोनेट करनी है, तो अगर आपकी किडनी मेरे बॉडी में डाल दें तो मेरा इम्यून सिस्टम उसको एक्सेप्ट कर लेगा, करेगा या नहीं करेगा? नहीं करेगा, एक्ट एज ए फॉरेन मटेरियल. तो दैट इज अ मेजर हिस्टोकम्पैटिबिलिटी कॉम्प्लेक्स. हिस्टोकम्पैटिबिलिटी कि आपके ऑर्गन कंपैटिबिलिटी क्या है, आपके डोनर कंपैटिबल है या नहीं, अदरवाइज आपका इम्यून सिस्टम उसको रिजेक्ट कर देगा. So these MSC molecules will catch that pathogen which is expressed on your APC. Antigen presenting cells will express one specialized protein. That protein is MSC. MSC. It's of two types, MSC1 and MSC2. So particularly if we talk about it, presentation is going for the helper T cells. So this MSC is MSC2 molecule. So if your antigen is catched by antigen presenting cells and presented by the MSC2 molecule, so it's always present to helper T cell. If your antigen is presented by MSC1 molecule, it will always present to cytotoxic T cell. Always remember that. If your antigen is catched by the presented by the MSC2 molecule, it will always present to helper T cell. If it's catched by the MSC1, तो किसको प्रेजेंट करेगा? बताओ, अभी तो बताया, cytotoxic T cells को, TC, this is TH, right? So these MSC2 molecules is going to present this pathogen to the helper T cells, but on helper T cells also there is a specialized proteins, and that proteins we call, which will send this pathogen, that protein we call TCR, T cell receptor. You heard about it, TCR or BCR. The receptor on B cell we call B cell receptor. TCR is T cell receptor. So this is the protein which is present on helper T cells, right? This which will sense this, which will be presented by this particular pathogen. Pathogen can be your bacteria, anything. For example, this is bacteria, right? अब TCR में बैक्टीरिया को तो सेंस कर लिया, प्रेजेंट हो रहा है अभी कोई एक्टिवेशन नहीं हो रही है एडैप्टिव सिस्टम, अभी जस्ट प्रेजेंटेशन चल रही है. अब दूसरी प्रेजेंटेशन क्या चल रही है ऑन हेल्पर टी सेल, देयर इज अनदर प्रोटीन, दैट प्रोटीन वी कॉल CD4. जब जाते हो लेबोरेटरी टेस्ट करने CD4 और CD8 काउंट करते हो ना, तो CD4 क्या रिप्रेजेंट करता है? एनीवन? कौन से सेल रिप्रेजेंट करता है CD4? एनीवन? CD4 you might heard about cluster of differentiations we call CD4, CD8, CD28, CD48. CD4 represent your helper T cell. CD8 represent your cytotoxic T cell. So CD4 is a protein on helper T cell. CD4. CD stands for cluster of differentiation. Cluster of differentiation means you are differentiating the cluster based on their marker, right? So helper T cells are many, right? So these are the cluster of differentiations by which you are differentiating. These are the helper T cells. CD8 is a protein, again it's a cluster of differentiations which is differentiating the cytotoxic T cells, right? So this protein and you know these particularly this is your TCR is catching this part bacteria, and your MSC molecules, these molecules should be combined with CD4. This is the presentation mechanism. One mechanism, primary mechanism. There is a post-stimulatory molecule as well. Again, I am repeating. Present कैसे हो रहा है? Presentation करने के लिए WBC के ऊपर एक specialized protein है MSC2, right? MSC2 ने किसको कैच किया हुआ है? एंटीजन को, right? एंटीजन को कौन सेंस कर रहा है? TCR. और जो आपका MSC2 है उसको कौन कर रहा है? कंबाइन किसके साथ हो रहा है? CD4 के साथ. अगर ये बाइंडिंग हो रही है आपकी, प्रेजेंटेशन मैकेनिज्म ये हो रहा है, देन देयर इज चांसेस ऑफ एक्टिवेशन ऑफ अडैप्टिव इम्यून सिस्टम. तो आपने बताया ना कि चिकन पॉक्स हुआ था तो बोला कि 6-7 दिन वेट करो, अपने आप ठीक हो जाएगा. क्यों ठीक हो जाएगा? क्योंकि 6-7 दिन ये सब प्रोसेस में लगते हैं बॉडी में, और एक बार जब आपके टी सेल बी सेल एक्टिवेट हुए तो उस दीज आर वेरी स्ट्रांग, राइट? स्ट्रांग इम्यूनिटी. कोविड में आपके कई लोग अपने इम्यून सिस्टम से ही ठीक हो गए, राइट? So particularly this is the primary presentation mechanism, but along with that there are a co-stimulatory molecule as well. Co-stimulatory molecule is your B7. You might heard about CD28. CD28 is on helper T cell. B7 is on your antigen presenting cell. Antigen presenting cell can be WBC, can be dendritic cells. Any cells can represent antigen to the helper T cells, right? So now, if these all एंटीजन को सेंस कर लिया टीसीआर ने, एमएससी2 को सेंस कर लिया CD4 ने, और B7 और CD28 भी सेंस हो गए, नाउ प्रेजेंटेशन इज कंप्लीट. अब नाउ your antigen is completely presented to helper T cell. Any doubt on this presentation mechanism? I am going to provide you the conclusion of QA. QA book है ना immunology की, इतनी बड़ी बुक है QA की, immunology. This is particularly on immunology. उसका summary बता रहा हूँ. So again I am repeating कि antigen कैसे present हो रहा है, और WBC is in this case is acting as a antigen presenting cell. और WBC उन एंटीजन को पकड़ेगा जो कि हैंडल नहीं हो रहे हैं, और कहां प्रेजेंट कर रहा है, अडैप्टिव इम्यून सिस्टम को. WBC के ऊपर प्रोटीन है MSC, राइट? MSC2 molecules will present the antigen to the helper T cell. If this antigen is catched by MSC1, then instead of helper T cell there should be a cytotoxic T cell, right? So MSC2 ये antigens will be sensed by TCR. TCR किसके ऊपर है, T cells के ऊपर, और ये MSC2 CD4 के साथ बाइंड होगा, और यहाँ पे B7 वाली प्रोटीन CD28 के साथ, तो ये कंप्लीट प्रेजेंटेशन हो गया. Is it clear? Now after presentation, now adaptive immune system is going to activate. अब उसको समझ आ गया कि इसने कुछ ना कुछ गड़बड़ की है, ये इम्यून सिस्टम जब पकड़ के लेके आया है, now it sense the complete presentation has been done. Now your adaptive immune system is going to activate, right? So how it's going to activate? It's going to process the transcription translation process. And by that process, it's going to release the molecules like interleukin 2, right? These released interleukins have the receptors on same cells. That kind of signaling we call autocrine or paracrine. Yes, अगर कोई cells कोई केमिकल रिलीज कर रहा है और वो केमिकल अगेन उसी सेल पे लग रहा है, तो उस that kind of signaling को क्या कहते हो, paracrine या autocrine? Autocrine. Right, so this is a classical example of autocrine signaling. So interleukin 2 will release by the cell and act on the same receptors which are present on the cell. Now it's going to under the process of clonal expansion. Before clonal expansion, there is a clonal selection. Clonal selection is क्योंकि आपकी बॉडी में हेल्पर टी सेल्स भी बहुत सारे हैं, तो कौन से हेल्पर टी सेल्स में आपका एंटीजन अच्छे से प्रेजेंट होगा, that only going to activate. Right? So now after activation, it's going to release some chemicals. That chemical will act on the same cells, and now they are going to differentiate. अब उन्होंने अपनी नंबर बढ़ाना है, तो नंबर कैसे बढ़ना है, यही इंटरल्यूकिन इसके लग रहा है, and then going the other process of clonal expansion. तो फॉर एग्जांपल आपके पहले हाइपोथेटिकली 50 थे, अब वो 1000 बन गए, आपकी आर्मी और आ गई, right? And the purpose is to kill this peptide. This is not handled by the innate immune system. And here is always a peptide, not a bacteria. Sorry, I have written bacteria, but it's a peptide. Why is peptide? Anyone? मैं bacteria क्यों नहीं लिख रहा हूँ वहाँ पे? Peptide क्यों लिख रहा हूँ? Peptide fragment will be presented, not the bacteria. ऐसा क्यों? कोई आंसर करो इसका? Anyone? I hope you understood my question. हाँ जी बताओ. इसलिए क्योंकि जब लड़ाई हुई इनिएट इम्यून सिस्टम के साथ, तो क्या बैक्टीरिया ही रह गया? उसका भी तो कुछ नुकसान हुआ होगा, उसका भी तो कुछ तोड़फोड़ हुई. दैट इज नॉट बैक्टीरिया, दैट इज नाउ योर पेप्टाइड फ्रैगमेंट, राइट? उसको भी कुछ डैमेज हुआ है, दैट इज नॉट कंप्लीटली हेल्दी. सो व्हेन देयर इज अ प्रेजेंटेशन, देयर इज ऑलवेज अ पेप्टाइड, वी कॉल इट पेप्टाइड फ्रैगमेंट, राइट? So once it's going to be clonal expansion is done, so it's going to release lot of chemicals like interleukin, interferons. Interferon is particularly for which kind of response? Quickly, antiviral या antibacterial या antifungal? Interferon. When we talk about interferon, antiviral. Interferon is antiviral, right? So this is your particularly how your immune system works, how immune system sense, how immune system present, and how your adaptive immune systems activate. But one more cell is left. Which cell is left? कौन से सेल्स रह गए अडेप्टिव इम्यून सिस्टम में जिसकी बात नहीं की which cells बताओ जल्दी दो तरह के सेल बोले थे अडेप्टिव इम्यून सिस्टम में टी एंड बी तो अभी बात ही नहीं की सो रहे हो क्या तुम लोग बी सेल राइट so B cell is going to activate. So the cytokines which is released by these helper T cells. Why we call it helper T cells? Anyone? Help कर रहे हैं किसको help कर रहे हैं B cells को भी तो helper T cell help कर रहे हैं किस चीज को help कर रहे हैं B cells को to produce antibodies, right? So it will helping to produce the antibodies, right? So B cells are particularly mature into plasma cells, and then plasma will convert into the antibody, and antibody against this peptide, against particular that pathogen. Your antibodies will be formed, right? Someone has talked about active and passive immunities. तो एक्टिव इम्यूनिटी में क्या करते हो वैक्सीन में क्या देते हो कि आपने उसी कोरोना वायरस को किल कर दिया नॉन पैथोजेनिक बना दिया आपकी बॉडी में डाल दिया अब क्या होगा आपकी बॉडी का इम्यून सिस्टम एक्टिवेट होगा by this process antibody will be produced against that one that is called active immunity active immunization और पैसिव में क्या हुआ ये एंटीबॉडी आपने उस पेशेंट में ऐड कर दी that is your passive immunity right so this is about your basic about your immune system so how you can do the experiments with respect to immune system is so I am going to talk on that any question up to this. So this already what I am going to cover is introductions, types, immune signaling pathway. So this already I have explained to you. What is the acquired immunity? What is the innate immunity? What is the primary line defense? What is the secondary line defense? What is the third line defense? Right? Then these are your innate immune signaling pathways, PLR dependent signaling pathways, NF-kappa B, MAP kinase, your TRIP, IRF3, and it's going to act on nucleus and going to result in transcription. These are antigen presentations which I already explained how antigens are presenting to the helper T cells or cytotoxic T cells. So this is a specialized mechanism by which your antigens will be presented to the immune system, right? So you can see that there is the MSC molecules. MSC are of two types, MSC1 and MSC2. There is a T cell receptors. There is your another co-stimulatory molecule. Co-stimulatory molecule is not only D7 and CD28. There are other molecules as well. Then these are the figures which I have taken from the Nature article, Nature Review article. So you can see that these figures already explain whatever I have explained earlier. So how antigen are presenting, and after that how it's going to do the release of various cytokines. This is all about your the innate immunity and adaptive immunities. This is about JAK-STAT signaling pathways we already discussed with respect to immune system. JAK is generous kinases inside the intracellular domain that is going to do the phosphorylation of tyrosine, and that is going to create your docking site for the STAT molecule, and STAT is going to do the transcriptions and going to release the cytokine. This is a PI3 AKT signaling. As I said, in the intracellular domain there is a PI3K instead of JAK. So phosphoinositide 3 kinases, it do the phosphorylation of PI3 2 into PI3, and then it's going to activate AKT, then mTOR, right? These are the mTOR signaling pathway. As I said, many molecules are under clinical trials and many molecules are in the market as well. So we are using these molecules, and these are the history of these molecules, how many of are in phase 2 clinical trial, phase 3 and phase 1 clinical trial. Now I am going to talk about with respect to immunotoxic agents. You might heard about there are some drugs, chemicals which is going to produce toxic effect to your immune system. So it can be antibiotics, it can be metals. Now metals are not being used in the dental, but earlier people are using. So it can also be immunotoxic, industrial chemicals, radiation, pesticides. In Punjab, there is a problem of pesticides. We are discussing about pesticide as well. There's particularly these Batinda regions. There is a lot of pesticide, use of pesticide, and that can also result in immunotoxicity. So, but human exposure to pesticide can be direct and indirect. Like maybe you are using these mosquito bites. How many of you are using? कोई यूज नहीं कर रहा, raise your hand. Only one or two. पंजाब में मॉस्किटो नहीं है. मॉस्किटो ज्यादा है. Okay, दिल्ली में ज्यादा है चल रहा है. मॉस्किटो ज्यादा है. So farmers are exposing to pesticide directly. Indirect exposure is whatever जो आप सब्जियां खा रहे हो, फल खा रहे हो, उसके थ्रू भी आप एक्सपोज हो रहे हो. वाटर, ड्रिंकिंग वाटर. So but does it mean कि हमें pesticide बैन कर देने चाहिए? So I am talking about immunotoxicity, but there are a lot of toxicity with respect to other organs as well. जब हम पेस्टिसाइड की बात करते हैं, तो कितने लोग मानते हैं कि पेस्टिसाइड को बंद कर दो यूज करना? Raise your hands. 1, 2, 3. बंद कर दे पेस्टिसाइड को यूज करना. और जो नहीं मानते उनका क्या आंसर है इसके लिए कि क्यों नहीं हमें बंद करने चाहिए? इतनी सारी जो टॉक्सिसिटी कर रहे हैं, वी हैव टू बैन दीस ऑल पेस्टिसाइड. बट अगर बैन कर देंगे तो क्या होगा? ये देख लो फिगर्स स्लाइड पे, ऐसी हालत हो जाएगी. प्रोडक्शन एंड क्वालिटी ऑफ फूड इज गोइंग टू डिक्रीज, राइट? Insect borne diseases are going to increase. Overcrowded. In India, already we have a limited facility in the hospital. You already seen in the COVID time. Overcrowding हो जाएगी, insect borne disease बहुत सारी हो जाएगी. So it's necessary and demand of time to use the pesticide, but you have to use it in a optimum way. So how to study the effect of drugs or chemicals on the immune system? That is your major part. If anyone are planning research work with respect to immune system, so how you can plan your work, right? I hope many of you are from master's background or PhD. So if you're planning to do the work with the immune system, so what kind of work you can do? So as I told you, first type of work you can do the computational studies, and under computational studies you can plan for immunoinformatics studies. Immunoinformatics, by which you can design the vaccine candidate with the help of computers, and you can do the complete thesis work. Some of my students have done immunoinformatics in master's. They have designed some vaccine candidate, and we have published that work as well. By computational, you can understand with the help of network pharmacology. You already, many of you knows about it or maybe working on. You can get to know about immune signaling pathways, how your molecule is going to activate or going to alter these immune signaling pathways. So that you can study by the network, and of course, when we talk about network, we are combining it with the molecular docking and dynamics, because network is not going to tell you about how your drug is interacting in the active site of target. To get to know about that, you have to do the molecular docking, and to check the stability, you have to do the molecular dynamics. So first one is immunoinformatics. So this is the work which we have done during the COVID time, particularly when there is a Omicron, the news of Omicron. So identification of vaccine candidate against Omicron variant of SARS-CoV-2 using immunoinformatics approaches. So as I told you, this work, all the work has been done by using freely available online servers. Nothing commercial software has been used to do this work. So we have designed the vaccine candidate against Omicron, and these are the steps. You can see that first step is we have performed the mutation studies, and then we have second step, prediction of antigenicity. So Vaccine is the server by which you can predict the antigenicity of that particular candidate. And step 3 is prediction of T and B cell epitope. We have predicted, and for the epitope predictions, we have done the cytotoxic T cell epitope, helper T cell, linear B cell, and conformational B cell epitope. And then 4 steps we have construct the vaccine candidate, right? Then 5th step, after constructing the vaccine candidate, we have checked the antigenicity, and all these work has been done by the computation. So allergenicity and the profiling as well, and then we have created the model, defined the model, and validated the model, and then we have done the docking. But in this case, docking is done with the protein-protein docking, because you want to see how your designed vaccine candidate is going to interact with the sensor. It's all like receptor. We have designed the vaccine candidate as a active immunity part. तो active immunity में क्या कर रहे हो आप? उसी पैथोजन का पर्टिकुलर पार्ट PAMP को you are designing in such a way when you are putting in the patient, it's going to stimulate your immune system. So with which you have to see the interaction. Whatever vaccine candidate we have designed, we have to see the interaction with the which receptor. So saw-like receptors, right? Pattern recognition receptors. So that is we call protein-protein docking instead of drug receptor docking. So that we have done, and then we have checked the immune stimulation profile as well. And this work has been published in silico pharmacology, not very big journal, but small work by using the all the three web servers, and we have published it in the in silico pharmacology Stranger Nature journal. Of course, its impact is too low, but work has been published, right? So you can also design in a similar way if any one of you want to do the project in the immunoinformatics. You can design the vaccine candidate, you can check the various parameters step by step by using freely web server, and you can publish it, right? So this is the one of the work which you can do with respect to immune system. Another work which you can do is how many of you heard about primary cell culture, primary animal cell culture? How many of you knows about it? Primary animal cell culture or cell lines. Not I will not use the word or. It's a and cell line. Cell line is different. Primary cell culture is different. So how many of you knows about it? Primary cell culture करना हो तो कैसे करोगे? Animal को लिया, ठीक है? Sacrifice किया, then to particular organs. So in case of immune systems, we have done the primary cell culture of thymus and spleen. Thymus is particularly for T cells and spleen is particularly for B cells. So my work was on specific immunity, not the innate part. So specific immunities I have worked on. So what we have done is you take the normal animals, you can't don't give any treatment, sacrifice the animal, collect the thymus, homogenize it, chop it properly in your media. We have used RPMI 1640 media, but if you don't have, you can do in PDS as well. You make the single cell suspension, filter it to 0.223 micron filter, and the filtrate you will get the single cell suspension. So first step which you have to do in such kind of work is after getting the single cell suspensions, you have to count the cells. You have to adjust the cell density. So how to adjust the cell density? Anyone? आपने सबने प्रैक्टिकल किया हुआ है अपने एचएपी वाली लैब में आपको मैंने हिंट भी दे दी ह्यूमन एनाटॉमी फिजियोलॉजी बी फॉर्म फर्स्ट सेमेस्टर में आपने कोई चीज यूज की है जिससे आपने सेल काउंट किए हैं क्या है वो जल्दी बताओ बी फॉर्म में तो सबने किया है ना क्या यूज किया है हेमो साइटोमीटर तो चैम्बर नहीं यूज किया आपने राइट So if you don't have in your lab, you don't have a cell counter. So you can count the cells by using that one. There you are counting the cells in the corner. वो square box होता है 4 You can count it and you can multiply it with the dilution factor. You will get to know how many cells are there, right? So that is the first step when you are working with primary animal cell culture. And I am talking about immune system. So what we have done is we have collected the thymus. We have collected the thymus for the T cells. लंच के बाद भी है लंच टाइम तो ही हो गया आप लोगों का आज पूरा दिन पढ़ा के जाऊंगा मैं तो इतनी दूर से आया हूँ दिल्ली से सुबह 4 बजे चला हूँ तो छोड़ूंगा तो हो नहीं पढ़ा के जाऊंगा प्राइमरी सेल कल्चर सो हाउ वी हैव डन द प्राइमरी सेल कल्चर अगर आप में से किसी को करना है जो भी फार्माकोलॉजी या किसी और भी स्ट्रीम के हैं इफ यू वांट टू चेक द इफेक्ट ऑफ योर ड्रग ऑन immune system. So this is the one of the way by which you can do this experiment, simple experiment. What you have to do is you have to collect the thymus. Thymus कैसे collect करोगे? Animal को sacrifice करोगे. हार्ट के ऊपर आपकी कवरिंग होगी. देखा होगा that is the thymus. So thymus you collect the thymus and chop it, homogenize it, mix them properly, right? Chop it in your media, cell culture media. You should have the cell culture media. And then filter it, filter करोगे and फिल्ट्रेट में जो आएगा आपका दैट इज अ सिंगल सेल सस्पेंशन। फिल्ट्रेट जो आएगा दैट इज योर सिंगल सेल सस्पेंशन, दैट इज कॉल्ड SCS, सिंगल सेल सस्पेंशन, SCS। अब इसका करने के बाद आपको क्या करना पड़ेगा? यू हैव टू एडजस्ट द सेल डेंसिटी। व्हाई एडजस्टमेंट ऑफ सेल डेंसिटी इज रिक्वायर्ड? बिकॉज़ फर्दर बाय these cells you have to design the experiment. So for each and every experiment, your number of cells can be varied. Like if you want to check the caspase activity, you need 3 million cells. If you want to do the MTD assay, you need 1.5 million cells. So that's why your adjustment of cell density is required. So how to do the adjustment of cell density? First you count the cells. अगर cells ज्यादा हैं तो आपको dilute करना है, अगर कम है तो क्या करोगे? सेंट्रीफ्यूज करोगे सस्पेंशन को, सेल विल बी सेटल डाउन, सुपरइंटेंडेंट को फेंक दो, उसके बाद उसको दोबारा से उसमें मीडिया डाल दो। पहले आपके उसमें 10 ml मीडिया था, अब उसको 5 ml कर दो, देन यू रिकाउंट इट, यू कैन डू इट। So you can adjust the cell density. Now, after adjustment of cell density, one test you have to do, that is a Crippen blue exclusion test. Have you heard about Crippen blue exclusion test? Crippen blue exclusion test. ये टेस्ट आपको करना ही है, सिंपल सा टेस्ट है, कुछ नहीं करना है इसमें। सेल लिए थोड़े देर अपने अपने ड्रॉप में, उसमें एक डाई डाल दी, उनको मिक्स किया, माइक्रोस्कोप के अंदर देख लिया, बस इतना सा काम। तो बहुत बड़ा टेस्ट है। डाई ली, उसके सेल्स के अंदर डाले, मिक्स की, माइक्रोस्कोप के अंदर देख लिया। अब why we call it Crippen blue exclusion test? Exclusion, why word exclusion is there? Because this is cell impermeable dye. कुछ डाई होती है सेल परमिएबल, जो सेल के अंदर चली जाती है, कुछ डाई होती है सेल इम्परमिएबल, जो सेल के अंदर नहीं जाती। तो ट्राइपेन ब्लू इज अ डाई व्हिच इज सेल इम्परमिएबल, इट विल नॉट एंटर इनसाइड द सेल इफ योर सेल इज नॉर्मल, राइट? इफ योर सेल इज नॉर्मल तो डाई विल नॉट एंटर इनसाइड द सेल। अब आपने मिक्स किया डाई के साथ सेल्स को, माइक्रोस्कोप के अंदर देखा, माइक्रोस्कोप में ब्लू ब्लू डॉट बहुत ज्यादा दिख रहे हैं, तो इसका क्या मतलब हुआ बताओ? सेल की इंटीग्रिटी, सेल मेम्ब्रेन की इंटीग्रिटी लॉस्ट हो चुकी है, क्योंकि मैंने आपको बताया दिस इज सेल इम्परमेबल डाई। इन नॉर्मल केस योर सेल मेम्ब्रेन इंटीग्रिटी विल नॉट अलाउ टू एंटर दिस सेल। पर अगर आपने मिक्स किया, माइक्रोस्कोप के अंदर देखा, आपको ब्लू ब्लू डॉट ज्यादा दिख रहे हैं, दैट मींस आपका जो प्रोसेस हुआ है इसमें आपने सेल जिंदा नहीं रहे, सेल वायबिलिटी हैज बीन लॉस्ट। तो जब सेल ही जिंदा नहीं रहे तो आगे के एक्सपेरिमेंट का क्या मतलब? तो आपको एक्सपेरिमेंट यहीं पे स्टॉप कर देना है, क्योंकि आगे आप एक्सपेरिमेंट लगा ही नहीं सकते, राइट? बिकॉज यू हैव डन समथिंग, या तो आपने सही से ये प्रोसेस नहीं किए, मैकेनिकल जो भी किए, तो उसमें आपसे कोई गलती की, जिसकी वजह से आपके मेम्ब्रेन की इंटीग्रिटी लॉस्ट होगी। तो ट्राइपेन ब्लू एक्सक्लूजन टेस्ट इज ए नॉर्मल टेस्ट बाय व्हिच यू कैन चेक दी वेदर योर सेल इंटीग्रिटी और सेल वायबिलिटी इज देयर और नॉट, राइट? इज इट क्लियर? बट आपने कितने लोगों ने सुना है एमटीडी ऐसे? रेज योर हैंड। एमटीडी ऐसे? हाँ, बताओ। टेट्राजोनियम, मिथाइल टेट्राजोनियम ब्रोमाइड, बड़ा सा नाम है, इतना तो मुझे नहीं ध्यान, पर दिस इज येलो कलर डाई। एमटीडी ऐसे यू माइट हर्ड अबाउट, वेरी पॉपुलर ऐसे which every lab is doing nowadays. MTD assay is telling you about the cytotoxicity of cell viability, right? So MTD is a yellow color dye, right? So yellow color dye, this is a cell permeable dye. Crippen blue is cell impermeable dye. तो MTD का मतलब क्या हुआ? ये नॉर्मल सेल्स के अंदर भी जाएगी, जो सेल्स की इंटीग्रिटी लॉस्ट है उसके अंदर भी जाएगी, दोनों में जाएगी। Is it clear? This is a cell permeable dye. Means it will enter inside all the types of cells, चाहे उसकी मेम्ब्रेन की इंटीग्रिटी मेंटेन है, चाहे टूटी हुई है, जो मर्जी है, ये अंदर जाएगी डाई। This dye will enter inside the cell. So inside the cells, in the mitochondria, there is an enzyme that is the succinate dehydrogenase. So succinate dehydrogenase will convert this yellow color dye into the purple color. अब क्या होता है जब आप एक्सपेरिमेंट कर रहे हो, वो यूवी के लिए प्लेट लेके जा रहे हो, आपके गाइड ने बीच रास्ते में देख लिया, अरे ये तो प्लेट तो सारी येलो ही दिख रही है, तो व्हाट डज इट मीन? अगर येलो दिखेगी प्लेट तो क्या मतलब होगा बताओ? सेल जिंदा है मर गए? मर गए। जिंदा होंगे माइटोकॉन्ड्रियली एक्टिव होंगे देन these cells will have the ability to convert yellow color into purple color. और जब सेल्स आपके कन्वर्ट ही नहीं कर पा रहे हैं तो दैट मींस सेल्स आर नॉट माइटोकॉन्ड्रियली एक्टिव, right? So MTT assays you can get to know that how many cells are mitochondrially active in the population. आपके पास cells हैं, how many cells are mitochondrially active? You can get to know by the MTT assays. You have to add this dye before 4 hours of the incubation. You can take the UV at 530 nanometers and you can calculate the percentage of cell viability. You can plot the graph, right? And you will get to know that what is the effect of your drugs on T cells. Major population is T cells in thymus. In spleen case, you have to, because spleen contains RBC as well. So when you are making a single cell suspension of the spleen, you have to do one more step. You have to add the ammonium chloride or any RBC lysis buffer as well, because you want to remove the RBC. So depending upon the organs, you can make the single cell suspension and you can do the experiment. So this is a well-known assay you will find with respect to MTD. So we have done with respect to thymus and with respect to spleen as well. This is your, and of course, MTD assay is not only assay which you can do. You can do further assay as well, depending upon your design of the experiment. So what we have done done apoptotic signaling. We have explored the apoptotic signaling pathway, complete signaling pathways of the thymocytes and splenocytes. So particularly how your molecule is going to activate the apoptotic signaling pathway and how your molecule is going to activate the apoptotic signaling pathway in T cells and B cells. So that experiment we have done. And of course, with respect to in vivo studies, also you can do with respect to immune system. If I talk about these T and B cells, so how you can do it? You do your experiments. Animals में आप डोजिंग दे दो, उसके बाद जब आप sacrifice कर रहे हैं, collect the thymus, collect the spleen, makes the suspension and perform the MTD assay, perform the ROS, perform the GS, perform the your caspases. You can do the DNA fragmentation. All these things you can do it, right? If you don't have a CO2 incubator, अगर आपके पास CO2 incubator नहीं है, so you can't do the primary animal cell culture. Primary animal cell culture, the basic requirement is you should have a laminar airflow unit, you should have a CO2 incubator, you should have a microscope. माइक्रोस्कोप आपका कंपाउंड माइक्रोस्कोप भी चलेगा, बट माइक्रोस्कोप होना चाहिए. ये तीन चीजें होनी चाहिए आपके पास. If you don't have a CO2 incubator, so cell will not survive, right? So how you will check the effect of your drugs or your chemicals on thymus or spleen? So in that case, you can do in vivo study. आपने animals को dosing दी, after that you have sacrificed the animal, you collected the thymus, you collected the spleen, and you have performed your assays. And assays, if you want to explore how your immune system is going through the process of apoptosis, you can completely do this experiment and you can explore the signaling pathway. And that is your nowadays, pharmacology is shifting towards signaling pathways, how your molecule is going to interact, what are the signaling pathways, so that you can do. And we have done all these assays, MTTs, oxidative stress, caspases, and these experiments we have done. Deltamethrin is one of the chemicals which is widely used. So how it interfere with your immune system and how your phytoconstituents or immunomodulators can accumulate its toxicity. So as I said, we have collected the thymus and spleen, we have prepared the single cell suspension, we have performed the MTD assays, we have performed the cascade activities, we have checked the oxidative stress level, we have done the immunophenotyping as well. And do you know about flow cytometers? Flow cytometer. Flow cytometer is the instrument by which you can check the physicals and chemical characteristics of your cells. So in flow cytometer, as its name indicates, flow cytometer. Cytometer means what? Cell, right? Cytometer. This is a flow cytometer. Flow cytometer is widely used in immunology or in your microbiology research. Flow cytometer. So cyto means cell, meter means measurement, right? So measurement of cells in flow system is called flow cytometer. If anyone asks you about flow cytometer, what is flow cytometer? तो आप सिर्फ वर्ड पे ध्यान दो, flow cytometer. Meter means measurement. Meter means measurement. Measurement of what? Measurement of cell cyto in flow system. आपके single cell flow करते हैं with the help of seed fluid. So you need a sample in a liquid phase. So in that one, when you are measuring the parameters of the cells, that we call flow cytometer. So we have a different lasers. You know, you have to be use different fluorochrome as well. If you are planning to do these experiments, you can get lot of information. So we have used this flow cytometers to do the phenotyping. Phenotyping means you can get to know that how many cells alpha T cell is going for the apoptosis, how many your cytotoxic T cells, how many regulatory T cells, so all these things you can do this. But if suppose you don't have a facility of flow cytometers, so what are the other way? Other way is by measuring the cytokines level, you can also get the information about which type of cells. Like alpha T cells mainly release your interleukin 2. So interleukin 2 you can measure by ELISA, right? ELISA किया है किसी ने? ELISA. ELISA techniques by which you can measure the absorbance based techniques by which you can measure the level of cytokines. So these experiments we have done, and this is another computational way by which you can explore the immunology. As I said, your molecules are interacting with your CD4, CD8, CD28, CD19. So there are immune cells have these cluster of different systems, right? Immune cells on its surface, some have CD4, some have CD28, some have CD19, like that. So with the help of computers, you can check the interactions with these molecules. Right, their proteins are available in the protein data bank, so you can do these molecular docking techniques by which you can check the interactions of your molecule with particular immune cells. Like आपको पता चलना है कि helper T cells के साथ मेरे phytoconstituent या मेरे molecules interact किस तरह कर रहे हैं. So you take the PDB of that particular cell or protein. Particular protein PDB you can take from the protein data bank. You can perform the interaction. You will get to know मेरा मॉलिक्यूल CD4 के साथ इंटरेक्ट करेगा अच्छे से या CD8 के साथ करेगा या किसी और के साथ करेगा. Right. So these we have done by molecular virtual dockers, and these are the results with respect to cytotoxicity which we have done. MTD assays. So in MTD you can check the effect of your molecules in a concentration and time dependent manner. So you can same plates you can check on 6 hours, 14 hours, 24 hours. So you can do it. These are the reactive oxygen species. Reactive oxygen species we have done by the fluorescence technique. By DCFX, DA is the one of the dye which is available on Sigma. So DCFX, DA. एक आपकी fluorescence based dye है. समझ आ रहा है नहीं? ज्यादा ये हो रहा है तो I can skip that part. DCFX, DA is one of the fluorescence based dye by which you can measure the ROS level. It enters inside the cell. Inside the cells there are esterases. Okay, so that will release your DCFH and then it can generate your fluorescence. Right? This is a Baxter plot. I hope you know about gel electrophoresis. How many of you knows about gel electrophoresis? किसी ने किया gel electrophoresis? So gel electrophoresis, by gel electrophoresis, you can measure the proteins. If we are doing vertical gel electrophoresis, there are a horizontal as well. Horizontal is agarose gel electrophoresis, where you are using the agarose for DNA, right? And for proteins, you are separating first proteins based on their molecular weight. Then you are transferring these proteins to the nitrocellulose membrane. Then you are incubating that one with the primary antibody, and then with the secondary antibody, you will get to know that whether your proteins are upregulated or downregulated. But it's a qualitative technique. To get it quantitative, you have to send the sample for the LC-MS. Then you will get to know that how much quantity is there. It's a qualitative, right? Mitochondrial membrane potential also we have measured by rhodamine is the dye and by using the fluorescence meter. So I'm not going to explain the results. These are the castages. Is the also we have measured because castages are the enzymes which do the degradation of DNA, and DNA fragmentation is the hallmark of your apoptosis. Glutathione we have measured with the OPT dye. This one is the flow cytometry result. This is the flow cytometry results where we got G0 by GS populations. You can get to know that how many cells are going to sub G1, and how many cells are going for the apoptosis. So you can see that by increasing the concentrations of the molecules, the cells which is going for the apoptosis percentages increase. So there we have used the propylenium iodide, right? These are the phenotyping we have done. As I said, on immune cells there are different markers CD3, CD19. So you will get to know that which particular type of T cell is going to decrease or increase by incubating your drug. So these we have done the phenotyping again with the flow cytometer, and then we have checked the effect of ion modulators. Ion modulators are required because you are going to explore the signaling pathways, and in bodies, there are multiple pathways. So how you would say that this is the only pathway which is responsible for this particular effect? There is a multiple pathways. So you have to do the experiments with inhibitors as well, right? So we have done with inhibitors, and in presence of inhibitors, when we have checked the percentage of apoptosis, we get to know that still there is a apoptosis. If there is involvement of only cascade dependent pathways, so apoptosis should be zero, but it's not zero. Still there is apoptosis. So what does it indicate? It indicates that there is an involvement of cascade independent pathways as well for this particular thymocyte simulation. And this work we have published in environmental toxicology, induced oxidative stress and mitochondrial cascade dependent signaling pathway in murine splenocytes. This is the thymocyte paper which we have published. Delta methylene induced and apoptogenic signaling pathway in murine thymocytes, exploring the molecular mechanism. So we are particularly investigated the molecular mechanism. Then we have checked the immunomodulatory role of your herbal molecules, your immunomodulated drugs, how whether they are going to reverse that or not. So again, we have first checked the by the computational techniques. We have done these with CD4, CD8 receptors and other receptors. Then we have checked with your drug molecule along with drug molecule whether it is going to reduce the toxicity or not. And these are our results on ROS. This is the GSS, caspase 3. Why caspase 3? Because caspases are many. Can anyone answer this question? Why I have mentioned caspase 3 only? Because caspases are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Why caspase 3? पढ़ाए बात में caspase का. Caspase 3 is the major caspases which will act on DNA, do the fragmentation of DNA. So we have, each and every experiment you can't do, but you should have a justification why you are going to measure this particular parameters only. Why you are not measuring caspase 9? Why you are not measuring caspase 10? If you have a good facility, you can do all, but it's always advisable. You have to design your experiment accordingly. So you know that the effect of these particular molecules on DLM induced of apoptosis we have checked, and these are the particularly on your particular molecules. So these particularly phenotyping also we have done in the presence of phytoconstituents. Piperine, what is the effect? And we have observed that at higher concentrations your piperine is going to reduce the DLM induced toxicity. These are cytokines, as I am talking about cytokines level, so you can do by the ELISA. So interleukin 2, if your molecule is going to decrease, so you can get an idea which type of T cells, that is a helper T cells or cytotoxic T cells, which cells are going to be mainly affected. So by measuring the cytokines levels, also you can conclude that which type of cells. As I said, helper T cells are mainly releasing interleukin 2, cytotoxic T cells mainly releasing interleukin 4. So by measuring their levels, you can get to know that, okay, which type of T cell population is affected by my molecule, right? If you don't have a flow cytometer, and if you don't have, you can fix the cell and you can send outside as well. There are many labs which can do the flow cytometer for you as well. You can fix the cell and can send them, right? So these are our work which is published in environmental toxicology and pharmacology about immunomodulatory role of piperine in delta methylene induced highly apoptosis and altered immune functions. And one more study which we have done with respect to immune system is we have checked the level of the molecules in immune organs, particularly in thymus, how much concentration of molecule is reached. In spleen, how much concentration of molecule is reached. If you want to do these kind of studies, you can do by using simple HPLC, right? So we have done by the HPLCs determinations of the concentration in mice plasma and immune organs. So So in immune organ, thymus and spleen, this is the particularly matter development and matter validation. You might everyone knows about. First, you have to develop the matter, then you have to validate, and these are our determination of delta matrix in immune organs by reverse phase HPLC. So these work has been also published in journal Acta Chromatographica, particularly where we have reported delta matrix in immune organs by simple reverse phase HPLC. Then in vivo studies, as I said, in vivo studies also you can design and you can do it. And this is the in vivo studies which we have published in Journal of Drug and Chemical Toxicology. This is the mechanism of delta matrix induced thymic and splenic toxicities in mice and its protection by the herbal molecule. Right? So these are the works which we have done with respect to immune systems. Ongoing work which we are doing is I just take 5 or 10 minutes. This is very interesting work which we are doing currently. Maybe it's interesting to me, maybe it's not interesting to you. But I just want to share what we are doing is we are going to develop a model, animal model, based on TLR. Why? Because you know, if for example you are planning to work on antibacterial area or antifungal or antiviral, you have synthesized some molecule or you want to check antibacterial effect of XYZ molecule or antiviral. So can you easily do the animal experimentation? Yes or no? आपको ethics committee approve कर देगी आपका protocol. In your protocol you have mentioned that because if you want to check antibacterial effect of XYZ molecule, so first you have to induce the infection in animals. Then only you can check the antibacterial effect or antiviral or antifungal. So can your ethics committee will easily approve your protocol? No, it will be rejected. Why it will be rejected? Ethics committee will reject your protocol if you have not taken one more committee permission. Do anyone knows what is that committee? IBSC, Institutional Biosafety Committee. So you need the approval of Institutional Biosafety Committee. Your institute should have the IBSC, and if your protocol is approved by IBSC, then you have to put your protocol in the IEC. It's not like that IBC has approved your protocol, so you can start the animal experimentation. No. To do the animal experimentation, still you need the IEC approval. But IEC will not directly approve your protocol if in your protocol you have mentioned you are going to inject the bacteria or you are going to inject this pathogen to produce such kind of infection, right? So it's a little bit, you know, the tedious task because IBSC again is on institutional burden as well. So what is the alternative way? So what we have thought of to resolve this problem is we thought that whenever you are getting any infection, जब आपको कोई infection होता है कोई body में जो हमने बात भी की, whenever any pathogen enter in our body, तो क्या हो रहा है? Immune system sense कर रहा है, sense करने के बाद ये signaling pathways, cytokines release or due to excess release of cytokines, these are doing the damage to the body, right? So why we don't have a such kind of system? और हम क्यों ना ऐसा एनिमल मॉडल बनाए जिसमें हम टीएलआर का एगोनिस्ट दे दें एनिमल को टीएलआर का आपने एगोनिस्ट दिया उसने उसके टीएलआर को पर्टिकुलरली जैसे आपको पता है टीएलआर3 वायरस को डिटेक्ट करता है आपने क्या किया सिंथेटिक टीएलआर3 एगोनिस्ट यू हैव इंजेक्टेड इन द एनिमल वो तो केमिकल है देयर इज नो नीड ऑफ अप्रूवल ऑफ आईबीएससी इज अ केमिकल व्हिच इज टीएलआर3 एगोनिस्ट आपने क्या किया एनिमल में टीएलआर3 एगोनिस्ट इंजेक्ट कर दिया so what will happen TLR3 agonist will activate the TLR3 receptors right yes or no और आपका जो वायरस एंटर कर रहा है बॉडी में वो क्या कर रहा है वो भी वही कर रहा है वो भी TLR को सेंस करके उसकी पाथवेज को एक्टिवेट कर रहा है so we are going to develop an animal model which is TLR based which can be used to screen preliminary screening of the molecules of antibacterial, antiviral, initial screening you can do in your institute without approval of IBSC. Understanding it? कर सकते हो नहीं कर सकते? Right? So this is the work which we are currently doing. We are developing the models and going to validate it. And इस मॉडल को आप फिर अपने एंटीवायरस के लिए यूज कर सकते हो. अगर आपके पास कोई एंटीवायरल मॉलिक्यूल पड़े हैं, बहुत सारा मैंने कई जगह बात की तो केमिस्ट्री पीपल हैव सिंथेसाइज्ड मैनी मॉलिक्यूल्स. फॉर एग्जांपल एंटीवायरल पे इसलिए काम नहीं कर रहा है क्योंकि एक्टिविटी करवाने में दिक्कत रहेगी एक्टिविटी के लिए फिर कहां सैंपल भेजें एनिमल की वो परमिशन नहीं मिलेगी so this is the one of the alternative which you can think of where you can, this is a development of and validation of PLR 3, 7 and 9 based model, and this is a poly 1 by C, which is synthetic dsRNA, and this is a PLR 3 approval. And to inject this in the animal, there is no need of ICSC approval. You can, of course, you need the institutional IAEC approval is required. Institutional Animal Ethics Committee approval is required. So you can do this development and validation. This is another PLR 7 and 8, which sends ssRNA. So this also we are going to develop. And then there is another one. Any questions so far? अभी तो बहुत स्लाइड है मेरे पास जाने में दूंगा लंच के लिए क्या टाइम हुआ अभी 1 बजे कौन लंच करता है यंग आदमी को तो लंच चाहिए भूख लगी है किसी को कितने लोगों को भूख लगी है लंच के बाद आओगे या भाग जाओगे पक्का प्रॉमिस कर रहे हो लंच के बाद कोई आपका नहीं है वैसे आ जाओगे चलो ठीक है सर बोल रहा है कि लंच कभी जाना है जब आप कंप्लीट करोगे कंप्लीट करने दीजिए लंच के बाद मुझे पता है आप लोग नहीं आओगे 100% एक भी नहीं दिखेगा क्योंकि हैवी डोज हो गया तो थोड़ा सा टाइम तो बस 10 मिनट दे सकते हो बस ठीक है 10 मिनट कोई क्वेश्चन मेन टाइम क्वेश्चन पूछो हाँ जी आप पूछो कुछ भी पूछो कि सर कहाँ से आए हो हाँ दिल्ली से और बताओ आप पूछो कुछ भी पूछ लो अरे इससे इम्यून सिस्टम से रिलेटेड पूछो कुछ आप पूछो कुछ ये तो बड़ा बहुत ट्रिकी क्वेश्चन पूछ लिया तुमने जिसे तुम्हें लगती होगी उतनी है और बताओ चलो बस 10 मिनट और ठीक है So these are the particular models which we are developing with respect to the immune system. So you can also think of if you are facing problems with respect to the screening of the molecules, you can think of alternative approaches. क्योंकि virus पे directly काम करने के लिए you need BSL-3 lab, right? So that is again a challenge. So you can develop this. And I told you didn't answer who won the Nobel Prize. नोबेल प्राइज आपको बता दो किस काम के लिए मिला नोबेल प्राइज पूरी इम्यूनिटी पर ली फॉरवर्ड पर्पस फॉर not central tolerance is already known so many years earlier central tolerance is earlier known peripheral immune tolerance can anyone in all tell what is peripheral immune tolerance and central immune tolerance बहुत सारी हमने इम्यून सिस्टम पढ़ लिया आप बताओ इसको अब तो इसमें भी लिखा हुआ है इन ऑन योर स्क्रीन यू कैन रीड इट एंड एक्सप्लेन इट व्हाट इज सेंट्रल इम्यून टॉलरेंस बताओ आई टोल्ड यू इन फर्स्ट एट द स्टार्ट ऑफ द लेक्चर द बेसिक फंक्शन ऑफ इम्यून सिस्टम इज टू रिकॉग्नाइज बिटवीन सेल्फ एंड नॉन सेल्फ If your immune system is not able to recognize between self and non-self, it will do destruction everywhere, right? And when your T cells are matured in the thymus, that time, if your immune system is sensing your own self, that will be go on the destruction, right? So, body have a abilities that your only non-self proteins or your self proteins will not be destructed. Your only non-self proteins will be destructed by the immune cell, not the self protein. So this is a central tolerance mechanism which is already known. Thymus में ये सब होता है process. Everyone knows about it. What these researchers have done, also these research have been very old. Nobel Prize अब मिला. Nobel Prize अब मिला इसका मतलब ये नहीं है कि उन्होंने 2025 में ये research की. They are working since last 20 years. 20-30 साल से काम कर रहे हैं. Right? So they have what they have particularly done. They have identified new immune security guard as a regulatory T cell. And regulatory T cells also playing a role to tell the immune system which one is self and which one is non-self. That is your regulatory T cell, and that is we call peripheral immune tolerance. And for this work, they got the Nobel Prize. And on this concept, currently you do the Google, more than 150 drugs are under clinical trial, and many drugs in the market for the anti-cancer use. You know about immunotherapy for the cancer patients? What it is? Immuno-oncology, lot of work is going. Checkpoint inhibitors. How many of you heard about checkpoint inhibitors? These all are the discoveries which are done based on this concept. What are immune checkpoint inhibitors? Immunotherapy, you don't know? In simpler way, immunotherapy for the cancer patients means, see, what are the cancer? Cancer is your own cells which are abnormally grown, right? So why your immune system is not able to recognize its non-self? Why it is so? Anyone answer? जब आपका कैंसर बन रहा है तो means जो आपका normal cell है, in that normal cell there is some alteration, right or wrong? So why that alteration is not sensed by your immune system? Sense होती तो immune system ही kill कर देता, right? Because there is lot of similarity between normal and cancer cells. That's why when you are starting a chemotherapy, why chemotherapy or anti-cancer drug have a lot of side effects, serious side effects. Reason is because the similarity between normal cell and cancer cell is too close. It's very difficult to differentiate. So whatever anti-cancer drug you are giving, that starts killing your normal cell as well, right? So immunotherapy means you are training your immune system in such a way that it will detect and kill the cancer. So there is no need of more, right? There is an immune checkpoint inhibitor as well. Due to lack of time, I am not going to explain, but immune checkpoint inhibitor. How many of you have heard about, because this lecture series is sponsored by Biotechnology Department, how many of you heard about CAR therapy? CAR therapy. In biotechnology, if you go to the Department of Biotechnology website, you will find many proposals have been asked for the CAR therapy. What is CAR therapy? Anyone? CAR T therapy. It's a chimeric antigen T therapy, T cell therapy. So what it is? In that one also what you are doing? In CAR T cell therapies, what you are doing? You are taking the immune cells of that particular patient, modulating that immune cell so that immune cells will recognize the tumor cell and do the destruction. That is your CAR T therapy. आप पेशेंट से सेल लेते हो, इम्यून सेल्स, उन इम्यून सेल्स को आप मॉड्यूलेट करते हो लैब में, मॉडिफाइड करते हो in such a way that these cells will able to identify the cancer protein. Or then again you inject it in the patient, and in that way your immune cells will recognize the cancer cell and start killing it. Read about CAR T therapy as well if you got a time. So just conclusion is immune system is a complex system, and there are a lot of signaling. We have discussed many signaling pathways. And more we learn about the immune system, more we amazed we are by its complexity. So still there are many questions remain unanswered with respect to immune system signaling, and a lot of scientists are working across the globe to explore this signaling, because immune system is one of the systems which is connected everywhere in the body. Right? If you talk about nervous system, if you talk about GITs, everywhere the immune cells are there, right? So these are the studies which we have published in International Journal of Review with respect to immune system. So these apoptotic signaling pathways with respect to thymus, splenocytes, your immunomodulatory roles, immunoprotective effects, and so on. So I would like to acknowledge all the departments who give me the funding: DST Sub, SRB and CRB projects, ICMR 2 projects, Sun Pharma, IIT Delhi, Sniper Release, Beat Mitra, History Faridabad, ISF Moga, and my current organization, the Pharmaceutical Sciences and Research University. And of course, I am always grateful to Bela College of Pharmacy for giving me the opportunity to be part of such kind of event. Thank you so much, and thanks to Kailash and thank you to all of you who have listened patiently. It's such a long time. I can understand you all are waiting for lunch, but still you have heard all the things. If you have any doubt, any questions, you can ask me. Anything. लंच के बाद भी आना है आप लोगों को। मैंने बोला अभी मैं उसको बोर्ड पे पढ़ाऊंगा। अभी एंटीमाइक्रोबियल वाला पार्ट तो रह ही गया, जरा भी लंच के बाद बुलाना है। लंच के बाद आ रहे हो ना? गुड, चलो थैंक यू।
लाइट्स ऑन करवा।
Thank you so much, sir, for enlightening session. Your talk has really deepened our understanding of the immune system and inspired us to further the amazing science behind human health. Now, to honor our respected resource person, I request Director Sir, Rahul Sir, and Ajay Kushwaha Sir, please come forward for the felicitation of Dr. Anup Kumar Sir as a token of appreciation and gratitude from our institute.
पहले फोटो फिनिश कर दो। अच्छा पहले सम्मान साइड में घुस जाता है कई बार। नहीं पता है सबसे कठिन काम इन लोगों का है सुनाना आसान पड़ जाता है, इतना हमारी मर्जी से बोल रहे होते हैं। एक ड्राइवर को लंबी यात्रा होगी ना वो कर सकता है, बट जो साथ में बैठे होते हैं वो बोर ही होते रहते हैं।
To conclude this wonderful session, I invite Dr. Ajay Kushwaha Sir for a vote of thanks.
On behalf of the organizing committee, I extend my heartfelt gratitude to Dr. Anup Kumar Sir from Vicharu, New Delhi, for his insightful and thoughtful provision of lectures on inside the immune system, guardian on human health. Your talk has truly enhanced our understanding of the interactions and mechanism of the immune system and its vital role in the maintaining of human health. I also thanks to DBT to supporting the popular lecture series and express sincere appreciation to the faculty members, students, and participants to their active involvement and enthusiasm. And I also thank you all making these sessions to enrich of memorables. Thank you, everyone.
Thank you. ये चल रहा है।